Ok, muchísimas gracias.
Good morning. Thank you very much for being here and not at the beach. We work at the University of Mexico, and I'm going to present to you some of the findings that we have about a new chemical form of enrofloxacin, which was finally done at this institution, the University of Mexico. I may be bold enough to remind you that developing a new drug, and especially a new antibacterial drug in Mexico or in the world, is very costly. And $80 to $100 million have been calculated. It's a high-risk investment, and I can tell you that from 10,000 molecules you can start with, you may end up with only one compound. So, as you can see, I'm old enough to say that time is running out, and we decided to recrystallize a molecule, which is a relatively easy way to find new entities that can have better pharmacokinetics and better biological activity. So we decided to test some fluoroquinolones. My wife and I worked with that for some years, and finally we found that enrofloxacin was a suitable one because it's very stable and has very low water solubility, and we wanted to increase that. Obviously, we needed to start with a high-quality raw material, and in this case, it is known that many manufacturing facilities worldwide are not at the top of the quality, and you end up with many compounds that have no biological activity or very low one, or have different chemical entities that will not give you the final results you're seeking. So it is important, and as such, it must be work in the manufacturing facilities, is that first you make a solvate. A solvate, for those who don't like chemistry, is the coordination with usually a metal, for example, sodium chloride or something like that. And then you recrystallize the reaction in different forms. It may be heat, it may be air, it may be dry, induction by many forms. Okay, this is an X-ray diffraction, and this one is the spectrum of enro-c. We call it enro-c because it's a new crystal, and if you overlap them, you can see that they are quite different, different enough to call it a new entity. You start with this yellow powder, which is high quality enrofloxacin, and after a chemical reaction, you end up with this white powder. The patent has already been established, and it is necessary for us to point out that enro-c is a white crystalline powder, very different from the yellow enrofloxacin high quality raw material. because we don't want to call it enrofloxacin hydrochloride dihydrate, which is a very long name, we decided to simply call it enro-c. And enro-c was defined chemically at Trinity College Dublin, where my PhD was, where I did my PhD, and at the University of Mexico. These details of this crystal have already been published in Acta Crystallographica for those who want to go into further details. Well, all that may be boring for you all, but then let's start with real pharmacology. And as you very well know, it is very important when you're treating an animal with fluoroquinolones to reach a Cmax, a high Cmax, is for us the most important pharmacokinetic variable. But of course, there are many other ratios that have been pondered or outweighed by pharmaceutical companies, like drug exposure, which is our area under the curve, divided by MIC, which according to, for example, the pioneers of this molecule, it should reach 120. Concentration dependent is Cmax divided by MIC, and usually it has been accepted that it must reach a value between 10 and 12. This is not for fluoroquinolones and should not be encouraged, especially now that the World Health Organization has called us to rationalize the use of antibiotics in veterinary medicine. Anyway, we have found, and this is important as well, that the bioequivalence of enrofloxacin is incredibly varied. You can find enrofloxacins that do not reach this value of 3.5 or almost 3.5 micrograms per milliliter. And sadly enough, the most sold, at least in Mexico and some parts in Latin America, enrofloxacin is this one, which is a mutant of enrofloxacin that, as you could guess, may give you very bad results, clinical results. But of course, clinicians don't think in terms of bioequivalence. They think in terms of bacterial resistance. Okay. When we started to test our enro-c, we found that it gave an incredible Cmax. And this is because our solvate, enro-c, is at least 20 times more water soluble than normal standard enrofloxacin. That allows a rapid absorption and an incredible Cmax. And this is a key feature that will give you the best results in the field. The generic ones are below, and including the reference one was reaching around 3 micrograms per milliliter. If you have a break point that has been accepted worldwide of 0.5 micrograms per milliliter, you need 10 times that amount to be within the Cmax divided by MIC ratio. And as you can see, you cannot reach that level with any of the enrofloxacins available. This is a bacteria that is already considered resistant. But if you use enro-c, you will usually find an answer because the ratio is much higher than the ones that are already in the market. What happened is that we didn't believe our own results. It was too good to be true. So we sent to this contract researcher in Canada our analog, which is enro-c. And the results that they gave us back is that our analog enro-c outperformed enrofloxacin and ciprofloxacin in a special setting that they have patented, Dr. Merle, which is a biofilm type of culture, in vitro assay. Anyway, we were very happy and decided to patent the drug in favor of the University of Mexico, of course, and so anything that has to be done with further developing this drug has to be dealt with the University of Mexico. We, by the way, are not selling anything, and we do not get any money out of any of these deals. So this is only our love for this new entity. Of course, we have done that many research and in many animals, dogs and cats and many. But as far as ruminant bovine is concerned, we have studied mastitis, endometritis, and some infections like bovine respiratory disease. Some of the pioneer work was done by Dr. Ismael Martinez. He's not in the room, I think. Haven't seen him, but he's not a researcher in another university in Mexico City. But it was difficult to publish this because of the results were outstandingly high in terms of absorption and in terms of concentration in target tissues. So one of the things that Dr. Ismael Martinez worked with was cytotoxicity of enro-c if you administer it through the intramammary route. He did some aquazombie marker assays in epithelial cells and macrophages and many other cells, and he found in the end that you can reach 300 milligrams per quarter, and you will find no toxicity of in epithelial cells or in macrophage or something like that. So finally, we convinced a pharmaceutical company to work with us, and there was an agreement, a licensing agreement between this pharmaceutical company in Mexico and the University of Mexico to work in huge dairies and start testing the drug. What we found and published in Journal of Veterinary Pharmacology is the efficacy in non-severe clinical mastitis. And, well, to our surprise, this was done by some students and physically, but it was followed by us. And you can see that efficacy was above 95%. This was an incredible result, and we were very happy. And it was repeated in many dairies, including the chairman of this Congress was involved, and other veterinarians as well. And after many, many glands being treated around 625, I believe, we found that efficacy was sustained at above 95%. The important is to stress to you all that you cannot use the normal enrofloxacin from pharmaceutical preparations because it has a very high pH and is very toxic. So I will not recommend to use any enrofloxacin unless it's enro-c, which has a pH of 6.5 in the commercial preparation already available in the market. When we tested against other drugs like enrofloxacin intramuscular from the reference preparation or ceftiofur intramammary from the reference preparation as well, we found an incredible difference. We have two enromastic, this is the new name, or commercial name the company gave to enro-c, but so I will name it either way. So enromastic was much higher, and bacteriological cure was also very high, especially in this group, and we were very happy to know that after 7 and 21 days, the original pathogen was completely destroyed. That what happened after that is that we even tested that after 3 cycles, you can see clearly how efficacy of enrofloxacin after intramuscular administration was decreased. And the same happened with ceftiofur, but that didn't occur with either enromastic 1 or enromastic 2. So potency was preserved throughout the life of the animal. And I'm talking about non-complicated mastitis. We carried out the studies for withdrawal time, and it's 3 days, either by chromatography or using charm or Delbo test. And as this veterinarian is telling to a colleague, this is the only fluoroquinolone which has an almost neutral pH and can be administered intramammarily to cattle. There are many products, and in Mexico monthly you can find even more, but there is only one enromastic in the world, and I think it's an incredible option for the clinician to work with. We're not happy with this final solution because we were treating only non-complicated mastitis. So we decided to tackle a new problem, which is Staphylococcus aureus chronic infection in cattle. According to veterinary clinics of North America, you should never treat these animals because they will contaminate other ones. But that is an opinion or a recommendation which has not been followed in Latin America, where usually the cow that is giving you more milk is the one that gets sick. So we started to prove long treatments with enromastic or enro-c to see if the cure rates improve. The ones that are accepted by, let's say, the FDA, which are around 50%, and it only mentions pyridomycin and ceftiofur. That was, let's say, one of the targets we wanted to reach. Of course, in many dairies, you can find this problem, and experience with this type of lesions told us that if we were going to use enrofloxacin for this treatment, we needed results in this type of cattle. And of course, after 10 to 15 days, we recover the mammary gland. So what we did is to carry out a study in which we identify bacteria producing biofilm-forming bacteria, especially Staphylococcus aureus. And after 10 days of treatment, we end up with a percent cure rate higher than the one obtained with Ceftiofur in the same number of treatments. This was very encouraging, so we decided why to fight against Ceftiofur. Let's join efforts. And I found this reference in which they sustained that there is a potentiating effect of enrofloxacin with the Ceftiofur. So we decided to test this hypothesis and isolated cattle which had pathogens resistant to fluoroquinolones. So what we did is 5 days treatment with enromastix, enro-c, which gave us 65% efficacy. And then, rather than stopping there and having this result as an end result, we added additional 5 days, twice a day, of Ceftiofur, and we got 100% cure. This has to be done with greater number of animals, but it was very encouraging to know that if you have this sequence, you will have high rate success of in curing these dairy cattle affected by resistant bacteria, which were characterized by this colleague of us, Dr. Edgar Alfonseca, which is not in the Congress either. But anyway, it's a great microbiologist at the University of Mexico. Then we thought it was convenient to attempt having further combinations with another elements, and we decided to test in biofilm-forming Staphylococcus aureus by Diana. Diana was a former student of us, and to try to destroy the biofilm. And encouraged by some of the review papers that I'm just showing you here. Okay, what happened is that Staphylococcus aureus, isolated from chronic cases of mastitis, had 100% ability to form biofilm. So that was the challenge. And what we found is that enrofloxacin, enro-c, plus dimethyl sulfoxide and chitosan were able to reduce the formation of biofilm. And so we went to treat many animals, and we found 100% clinical cure, 80% bacteriological cure with this new formulation. We are now pursuing another formula because we believe that it is possible to obtain 100% bacteriological cure, but that'll be for the next Congress. Anyway, you start with lumpy glands like these ones, easily seen by anybody that it is affected. And after identifying that it was Staphylococcus aureus, the causative agent, we started treatment. In 5 days, you can appreciate the difference in the mammary gland, how it is softened, the surface. And if you pursue the treatment, you end up with a mammary gland that is soft, has no bacteria that could be isolated in most of the cases. So it is a very encouraging result, but we want to tell you that there are many references in the formal literature in which they state that there is synergy between phytoelements and some of the antibiotics that we use. Then, changing the subject, we are now trying to reach a point in which we have enough evidence to start working with enro-c. One of the persons that pioneered this work is Dr. Jesus Mendoza, which is sitting over there. And he carried out studies of pharmacokinetics, comparing the reference one and some of the supposedly generic enrofloxacins. And you can see easily how a generic enrofloxacin has a very low Cmax, a flat curve, because of the reasons I already told you. When you compare the pharmacokinetics of serum profiles of standard enrofloxacin, it's good enough. It reaches 3 micrograms per milliliter, more or less. But in our case, enro-c reaches 7 or nearly 7. So when you do the pharmacokinetic program, we have enro-c with high Cmax value. And this is very important in terms of clinical efficacy. Reviewing the literature, these authors show that if you want to have concentrations able to destroy bacteria that already need mutant preventive concentrations nearly of 0.5 micrograms per milliliters for enrofloxacin, then you need a higher dose and a high bioavailable enrofloxacin. So translating the pharmacokinetic data into Monte Carlo simulations, we found that if you have, for example, Mannheimia hemolytica and E. coli resistant at the level of 0.5, you will reach enough concentrations to have high efficacy with these high doses of enro-c. But if you attempt the same with the standard enrofloxacin from reference preparation, you will reach only 20% the target attainment, which is Cmax divided by MIC equals to 10. This encourages to attempt the same Monte Carlo simulation, but now against Leptospira. You may be aware that enrofloxacin is not recommended to treat leptospirosis, but we took that as a challenge, considering the high Cmax obtained by enro-c. And again, in this limit, we found that enro-c is capable of destroying Leptospira. to test that hypothesis, and I want to emphasize that it's only enro-c the one that can kill Leptospira. We, with Dr. Mendoza again, did some studies in which treated bovine leptospirosis with enro-c, and again with high success rate. So the same happened with sheep. Now, just to show you, before we came here with Dr. Posadas, the chairman of this Congress, some clinicians brought these calves. And these calves were very sick, and they were already dying. As a matter of fact, you can see the difficult breathing of the animal, and after necropsy, bacteriological samples showed that it was Klebsiella hemolytica. This is a common pathogen in hospitals in humans, but this is the first time I hear about it in the field. So what happened is that these animals, 3 calves, came to the University of Mexico, and they could hardly stand, and they had nervous movements and end up with epileptic fits due to meningitis. And of course, he was put down humanely. But the point is that we needed to do something with the rest of the calves. So we injected enro-c. And with a single injection, you could see that this was sent to us by one of the vets in the field, and it shows how temperature is becoming almost normal, and it's already suckling and behaving normally. Enro-c was then produced because we do it at the lab scale. Now the pharmaceutical company is trying to increase its production, but anyway, these animals were previously treated with tulatromycin, with Septifur, and other drugs, and with no success at all. So this is the type of challenges that we are now testing, like this one. This is enrofloxacin from enro-c. Enro-c, but in polymer base, so we can mimic the results of the enro reference of long-acting, and our enro-c. This is a logarithmic scale, so you can see that it doesn't show too much, but it reaches almost 8 micrograms per milliliter. We tested this to see which one was the best pharmacokinetic ratio, AUC divided by MIC or Cmax divided by MIC. So we tested that in the clinic. I just want to stress the PK, the pharmacokinetic parameters, which are incredibly superior for enro-c as compared to the enro reference of long action, and our own enro-c made long acting. So we can test it to see which parameter was the best. For that, we made a composite table in which we could organize the severity of the respiratory disease, because reading about this subject, we encountered that the best clinician in this hall will have at least 40% error in detecting whether it was severe respiratory disease or not. But after taking into consideration all these clinical signs, we found in a later study that we will publish, this is very recent, that the error was reduced to around 10%. Okay. Anyway, what happened is that if you sum all the clinical signs, when you use enro-c, they decrease sharply. And in 3 days, you have most of the calves resolved from a respiratory disease. This is part, when you see this problem, of course, if you sell tulathromycin or gamithromycin or one of those macrolides, you tend to recommend its administration before anything happens. This is a different approach. These are animals that are already sick and they are very ill, and they have to be treated with a drug that kills bacteria rather quickly instead of slowly like macrolides, which are great. I am not against macrolides. I'm just saying that it's a different clinical scenario. Well, finally, we tested enro-c against endometritis, considering that it has a pH almost neutral and is not toxic to endometrial cells either. Well, we thought of tackling this problem, which, according to literature, is very costly, too high perhaps. And based on this review by these authors, we decided to test enrofloxacin from enro-c in this setting, clinical metritis, which is around 20-25 days, depending on the clinician. And at this stage, the possibilities of the cow to be spontaneously cured are sharply decreased. Then we, based on Sheldon, which classify the secretions in this grading, we just tackle the 3 most affected grades of endometritis. What happened is that you may think that only prostaglandin is enough to treat these animals, and that may be true, but once they have clinical endometritis, that is not, let's say, accepted by many specialists, clinicians that work with reproduction. Dr. Raul Garcia Moreno was a student of us who started or helped us with this project, and we started with many cows, Holstein mainly, and in another state near Mexico City. We compare our results against the gold standard of cefepime from commercial preparation. And in both cases, we injected cloprostenol as an aid, because by itself, in some of the references, it has been said that works as well as antibiotics, which in the field I may question this. Anyway, what we have found so far is that percent efficacy with cefepime is good enough, but we are having much better results with our enro-c. Okay, follow up. We encounter, treatment intervals to first estrus was improved, number of insemination to gestation, and number of open days were reduced. This hasn't been published yet because we still want to gather enough information. Okay, this product may be released in the short term in terms of a single application because it was just a one application. And I thank you for your attention. And do we have time for questions? No? Are you still awake?
Thank you much for your presentation. It's very insightful. Some of the enrofloxacins in some countries are registered with flexible dosing, so you have 2 milligrams, 5, 7, 10, or 15. Is that applicable to this product?
Yes, we can. We recommend the highest dose possible to reach the Cmax. And for example, in dogs, we can reach 10 to 50 milligrams with incredible clinical results and no side effects. That's very important. So when you start recommending lower dose, you may lower the results, clinical results. So for me, it's better 10 and above. Of course, the enrofloxacin is open to negotiation with the University of Mexico and the final products as well with the pharmaceutical company. Okay.
Thank you, Dr. Sumano.
She's my wife. Okay, next presentation. Está aquí el doctor, o la doctora. Anastasia Lisuzzo is going to present evaluation of the healing process of long lesions following treatment with florfenicol and meloxicam in veal calves affected by bovine respiratory disease during one production cycle. To forward, to point out something.
Okay, thank you so much.
Okay.
Okay, good morning, everyone. Thanks for the presentation, and thanks to all of you to be here. Today, I will show you the results of this study about treatment based on florfenicol and meloxicam for veal calves affected by bovine respiratory disease. Bovine respiratory disease is one of the main diseases in cattle livestock, and the gold standard test for performing a diagnosis is a necroscopy examination. However, diagnosis in vivo is usually based on clinical signs, and these clinical signs that can be both systemic, like rectal temperature, or respiratory signs, are used to establish several kinds of clinical scores. And the two most used clinical scores are the Wisconsin score and the California score. The California score is based on presence or absence of these clinical signs, whereas the Wisconsin score is usually based on the severity of the clinical signs that we can observe. However, lung ultrasonography is a recent tool that started to be used for the diagnosis in vivo for BioD. And this technique showed an higher sensitivity and specificity compared to clinical examination to perform an in vivo diagnosis. And this higher sensitivity and specificity help us to improve the identification of the animal's health and consequently reduce the antimicrobial use for unnecessary cases. So animals that show clinical signs, but they are associated from an upper respiratory disease tract. And consequently, with lung ultrasonography, we can also follow the animal to evaluate the healing process of the lung after a BID case. The aim of this study were to evaluate the bovine respiratory disease over the production cycle of veal calves, use a treatment of florfenicol and meloxicam, and evaluate this treatment with clinical and ultrasonography parameters, and after evaluated the healing process of the lung after the treatment. For this study, we enrolled 84 crossbreed veal calves, and this animal, at the beginning of the restocking period, had an average age of 30 days, and we followed this animal with clinical examination and lung ultrasonography twice a week for the first 2 months of the production cycle, and after we continue to follow animals with the same kind of examination weekly during all over the production cycle. So during each examination, we collected the data about clinical signs, so rectal temperature, cough, breath characteristics, nasal and ocular discharge, fecal consistency, hair position, and sensory states. And we used all these clinical signs to evaluate the 2 previously mentioned clinical scores, California and Wisconsin clinical scores. Furthermore, we performed the last ultrasonography with the Raminsky Blue ultrasound scanner, and we performed this ultrasonography on both lungs, left and right, of the cranial, middle, and caudal region for each animal. So we used this ultrasonography image to evaluate the animal with 2 different kinds of scores. The ultrasonography score is a well-known score for evaluate the animals and perform a diagnosis of bovine respiratory disease with lung ultrasonography. And this kind of score is based on presence and absence of lesion, and evaluate only 2 types of lesion, the cometail and consolidation. This score goes from 0 to 6, and usually animals are considered as affected by bovine respiratory disease when the score is equal or greater than 3, so the animal shows at least a lobar consolidation. The second score is the lung lesion score. That is a recent kind of score that we started to use. This kind of score obviously evaluates presence and absence of lesion, but also the several types of lesions that we can observe in pathological lungs. So we have cometail, hepatization, fluid alveolograms, fluid bronchograms, pleuritis, and a combination of this kind of lesion. And in this kind of score, we evaluate the several types of lesion in each region. So 2 cranial region, 2 middle region, and 2 caudal region. We sum the score of this kind of lesion for each region and obtain this lung lesion score. Consequently, we saved all the images, and after we also performed a measurement of this lesion for each region, and we measured the area of each lesion for each region and the thickness of the lesions. Regarding the treatment, we decided to treat animals when they show at least an ultrasonography score equal or greater than 3, and also if they showed a lesion in the cranial lobe greater than 3 centimeters of thickness. In these cases, so we proceed with the treatment with florfenicol and meloxicam, and we systematically follow the animals after the treatment. whereas the animal that never receive a treatment during the production cycle were included in the control group. Consequently, we continue with statistical analysis based on mixed models. Regarding the enrollment of the animal in the treated group, most of the animal, around the 90% of them, were enrolled during the first month after the arrival, so the beginning of their stocking period, whereas we completed the enrollment during the first 2 months after the arrival. Regarding growth performances, there were no difference in average daily gain, live weight, and cold carcass weight between treated animals and control animals. We considered this lack of difference due to the fact that most of the enrollment were at the beginning of the production cycle, and consequently, the animals had the time to recover from the biodecays and reach from the growth performances the control group. So we can observe the efficacy of the treatment due to prompt treatment and an early diagnosis, also for the lack of difference in growth performance between treated and control animals. Regarding the comparison between treated and control animals, there were no difference at the arrival, no in clinical score, both Wisconsin and California score, none in ultrasonography score and lung lesion score, or in total consolidation area. As you can see, at the beginning of the dry period, all the animals showed at least some kinds of lobular consolidation of about one square centimeter of consolidation. So it's important to note that not all animals that show some degree of consolidation can develop bovine respiratory disease, because in this case we have both control and treated animals. There were no difference also at the end of the production cycle in clinical score, ultrasonography score, lung lesion score, and total consolidation between treated animal and control animal. Obviously, at the treatment day, the treated animal showed a higher clinical score, both Wisconsin and California, a higher ultrasonography score, lung lesion score, and total consolidation area. And at the treatment day, the total consolidation area was about 30 square centimeter in the treated animals. Regarding the comparison between lung ultrasonography and clinical examination, what was observed was that the animal at the treatment day showed the highest ultrasonography and lung lesion score, indicative of bovine respiratory disease, but despite this, animals showed an increase in clinical score, both Wisconsin and California, but this increase in clinical score were not indicative of bovine respiratory disease. Instead, the clinical scores, both Wisconsin and California, showed the peak indicative of bovine respiratory disease 5 days later compared to lung ultrasonography. So the use of lung ultrasonography was able to anticipate of about 5 days clinical signs in order to perform the diagnosis of bovine respiratory disease. Regarding the lung healing process, after the treatment, the animal showed a significant decrease in the ultrasonography score after 3, 5, and 11 days. And after the 5 days, the animal showed an ultrasonography score under the cutoff indicative of bovine respiratory disease. If we consider the lung lesion score, the treatment, we have a significant reduction after the treatment. After only 1 day, 5 days and 7 days. And also in this case, after 5 days, the lung lesion score was under the cutoff indicative of bovine respiratory disease. The difference between the different time of reduction between these two kinds of score is probably due to the fact that, as I said, ultrasonography score is based on only two kinds of lesion, cometail and consolidation, whereas the second score, the lung lesion score, is based on several types of lung lesion that we can observe in pathological condition. So probably the difference between 3 and 1 day for the reduction is due to this difference. Regarding the total consolidation area, we can observe a reduction after the treatment in this area. After 1 day, 5 days, and 9 days, as you can see, after 1 day, we can go from 30 square centimeter to around 15 square centimeter of total area of consolidation. And also, as for the total consolidation area, also the thickness in the lesion of the cranial lobe, we can observe this reduction after around 3 days in both lungs, so right and left lung. Consequently, we can say that the treatment efficacy with florfenicol and meloxicam was about more than the 90%, whereas we have a little less than the 3% of fatality cases and chronic cases. So in conclusion, we can affirm that the first month after arrival is the critical phase with the higher risk of bovine respiratory disease for veal calves, and that a systematic lung ultrasonography is able to perform an early detection of bovine respiratory disease, anticipating of about 5 days clinical examinations. And the early identification, so the early diagnosis, ensure a prompt treatment, and consequently, the animal had more chance to recover from this disease, and so there were no difference in growth performance. And regarding the healing process, the treatment with florfenicol and meloxicam showed a significant improvement in lung lesion score and total consolidation area after 1 day, whereas in ultrasonography score and lesion thickness after 3 days from the treatment. Thank you for your attention. Thanks to all the members of the group that cooperate for this study, and to the company Seva Salute Animal and the Dramiski for the ultrasonography scanner. Thank you so much.
Any questions? No? I have a couple of questions. Okay. I have a couple of questions. First is, how long does it take to examine a calf with your ultrasonograph?
Yeah. So depends by the experience of the vet that perform this kind of analysis, but when you are trained, you can use like 1-2 minutes after that the animal is stopped, so
restrained.
is captured. Yeah, is restrained. After that is restrained, 1-2 minutes to perform a complete lung ultrasonography of both lungs.
And you need gel to smear
No,
it?
is in my opinion, is better ethyl alcohol at 90%, because gel creates some kind of problem in farms. So in my opinion, is better alcohol.
Okay, and another question is, what is the cost of the machine?
Oh, the machine very well depends by your agreement with the company. Is some millions, around around 10,000 of euro in Italy, around this one.
10,000 euros in Italy.
Yeah.
That's a lot of money in Mexican pesos.
Yeah, I mean.
Okay, have you any other question or something to clarify with?
Good morning, doctor. Thank you for the information. Do you have any data or any investigation about the cost, the treatment cost against the benefits of results?
Okay, very well. We try to check also the benefits, so the cost for the meat. But if I remember correctly, there were no differences between the treated animal and the control animal in the comparison between treatment and benefits. So they were equal at the same.
In profit is the same?
Yeah, around the same.
Okay, thank you.
Thank you.
A ver, ¿le vamos a dar esto? Okay, if there...
Hello.
Hi.
Congrats for the presentation. Very nice presentation. I have a little question. Do you have any data on recovery rates or lung lesion recovery on calves that are already showing clinical signs? In my experience, the ultrasound recovery rate is not in 2 or 3 days. It could take up to months. So.
Okay, so regarding the healing process of the lungs, depends about severity, treatment, when you performed the treatment, because if you performed an early diagnosis and consequently had a prompt treatment, you have a lower risk that the lesion continue to be chronic, okay? Whereas if there is a retarded diagnosis, a later diagnosis, is the risk that you have chronic case is more higher. But in this study and also in other studies that we performed regarding treatment of bovine respiratory disease, we observed that in 3 to 4 days, you can see a real improvement in lung lesion if you performed an early diagnosis of the bovine respiratory disease.
Thank you.
Okay, this was an interesting observation because in humans, you treat a pneumonic patient and the lesions you see in the x-rays are still there after 15 days. So what the doctor is telling us is that with ultrasonography, you can shrink the lesion within 3 days.
Yeah, you can see this reduction. Obviously, we need also to consider it as a perfect lung, so without any kind of lesion, is impossible in our animals, so there will be always some kind of lesion. The important thing is that the lesions are really small, doesn't develop in bad cases.
Okay, thank you very much. We appreciate your great effort in coming here. I was told that we have 5 minutes for coffee and for their biological needs. So see you in 5 minutes before, if possible. Huh? Okay. Um, I think we need to continue the presentations, and in this particular occasion, we're going to hear what Dr. Itzcoatl Aquino is going to tell us about the pharmacological perspective on the use of Benzyl-penicillin G in production veterinary medicine in Latin America. This is a very important subject because in our view, penicillins are being not correctly used in Latin America.
Thank you, Doctor. Good morning. I'm going to talking about pharmacological perspective on the use of Benzyl-penicillin G in production veterinary medicine in Latin America. Okay, I'm going to start with some generalities about Benzyl-penicillin G or BPG. It's one of the most used antibacterial drugs in human and veterinary medicine. An analysis should be made to ensure the adequacy of its use based on rational fundamentals of pharmacokinetics, pharmacodynamics, and is a time-dependent antimicrobial. The beta-lactam ring is present in all beta-lactam antimicrobials. Benzyl-penicillin G is the origin and precursor of the historical development of the beta-lactam antimicrobial series. Resistance to penicillin occurs mainly through the inactivation of the beta-lactam nucleus of the drug, and bacteria that produce beta-lactamases may exhibit cross-activity. Streptococcus species tend to show standard sensitivity to BPG worldwide, and the BPG breakpoint assigned is 0.4 micrograms per milliliter. And the beta-lactam antibiotics have lower action than other bactericides. They don't have a post-antibiotic effect, and the dosing regimens should consider their PK/PD relationship. There are 4 BPG salts. We have BPG sodium and potassium. They have fast absorption and quick removal, and with a dose of 50,000 international units per kilogram, they difficult compliance with a PK/PD relationship. And if we want the therapeutic effect, we have to administrate continuous dose to maintain the concentrations above the breakpoint. Other salt is BPG benzathine. This has slow absorption and slow elimination, and with administration of 20,000 international units per kilogram, it has low concentrations, but with a long withdrawal time, maybe more than 20 days or more. And BPG procaine has slow absorption and slow removal with 20,000 international units per kilogram. Has antibacterial concentration for almost 24 hours, but we can have sometimes with no efficacy therapy between the applications. Procaine BPG is the best penicillin, but what's the best dose? If we consider the breakpoint with 6,000 international units per kilogram, the concentrations in plasma are under the breakpoint. With 20,000 international units per kilogram, the concentrations are above the breakpoint, but not all the time. And with 65,000 international units per kilogram, the concentrations are above the breakpoint all time, but this could be so expensive. We can modify the administration interval to improve the 20,000 international units dose. When we administrate each 24 hours, we can have periods without antibacterial activity, only acts against sensitive bacteria and does not act against less sensitive bacteria. And when we administrate each 12 hours, we have continuous antibacterial concentrations, ensures the PK/PD ratio against sensitive bacteria, and acts against less sensitive bacteria. It's important to remember that MIC may vary depending on the microorganism. A microorganism can have MIC with a wide range. If the MIC is greater than the antibiotic concentrations at the site of infection, it will not be effective. The recommended dose in intramammary is 600,000 international units per affected quarter, once per day for 5 days. However, some products in Latin America only recommend the half of the dose on the label. And another recommendation for some products is the combination of BPG plus aminoglycosides. It suggests that the use of the combination of BPG and aminoglycosides does not increase the effectiveness of the treatment, and no synergistic effect was observed in vivo on Streptococcus vivaris. After the parental application of BPG, the concentrations in milk are only effective against very sensitive bacteria, and the PK/PD relationship is only achieved for very sensitive bacteria. Can streptomycin improve the effectiveness of BPG? If we consider the MIC for streptomycin susceptible bacteria is 5 microgram per milliliter, after the parenteral application, the concentrations in milk are lower than the MIC, and there's no synergistic effect for the treatment of mastitis when administered parenterally. Does the combination BPG and streptomycin have therapeutic advantages? There is information about the inefficacy of this combination, and it's better the use of each drug alone. And if we consider the milk withdrawal time, after the administration, the parenteral administration of BPG, the recommended withdrawal time is 7 days. However, some products in Latin America only recommend since 72 hours until 4 days. On the other hand, some of these products include Benzyl-penicillin G, and this is a drug with a long, has a long withdrawal time. Benzyl-penicillin G is authorized in human medicine only to treat some diseases caused by hypersensitivity pathogens. In this table, we can see some examples of banning benzathine BPG according to various regulatory agencies. And as we can see in Australia and the United States, the combination with streptomycin and dihydrostreptomycin is banned. Some recommendations for Latin America are only use protein BPG in production animals, use a minimum dose of 20,000 international units per kilogram every 2 or 24 hours for 4 to 6 days, and this information must be mandatory on labels. Do not register or use other drugs with BPG protein unless there's evidence of stability and efficacy, and adjust the withdrawal times of protein BPG according to its pharmacokinetics, 7 days in milk and 15 days in meat. And this information must be mandatory on labels. Thank you for your attention.
Okay, there should be a lot of questions because in Latin America we're using many products that are combined with the streptomycin, neomycin, and many other combinations are available. And we should use penicillin only by itself and at the doses recommended by Dr. Aquino. So is there any question? Are there any questions, or have you got any experience to share?
I have one. Yeah, just as you told, for example, in my country, in Colombia, it's very common the use of penicillin, streptomycin, and dexamethasone, the same bottle in the same shot. So that's the most common drug used for metritis and also for toxic mastitis. So why do you think is the reason for the labs to produce those mixing shots and bottles if they know this information?
These combinations are so popular and it's common, recommend from one vet to other vet, but if we search the information published, the information says there's not enough activity when we use both. Maybe for me, it's so attractive, combine a lot of things. And that's the reason for me, and maybe that's the reason.
Pharmaceutical companies have told us that if you buy it, they produce it. So that's the main reason.
So I can probably share some experience. I do actually work for a pharmaceutical company as well, but I'm from New Zealand. Procaine penicillin is very widely used in New Zealand and very successfully used. We are using those doses, the high doses for injectable procaine penicillin. It's very successful. For mastitis, it's the most common drug that is used for treatment of mastitis. And again, it's very, very efficacious for the right bacteria. Strep uberis, extremely good. Even Staph aureus. So the sensitive Staph aureus, it works very, very well against. The resistant ones, even with a high enough dose, it will actually still work quite well. It will overcome the beta-lactam resistance with a high dose. But we use 1 gram of penicillin. So you're recommending 600. I know you're saying 300 is the dose that's in most of the products. We are at 1,000 milligrams. But good efficacious, good efficacy at those doses.
Thank you very much for sharing this information with us. If you have those words written in some paper, it will be very useful for all Latin American countries to know them, because it's a marvelous drug, but it's being ill used in Latin America. Thank you very much.
Yeah, no, I agree. It is actually it's an older drug, of course. I mean, it's the first antibiotic, and I guess we think sometimes the more modern drugs must be better, but in fact, in some cases, it's still the best drug there is. So, yeah.
If you can give us later your email, I can write you down so I can share. Even if you just write me an email, that'll be enough for our regulatory agencies to see it and to see that we are trying to avoid the use of low doses of penicillin in Latin America.
I can talk later. Yeah.
Thank you. ¿Algo más? Something else? No? Okay. Thank you very much, Dr. Aquino.
Thank you.
The following presentation is a quick one. Is the evaluation of an alginate combination with enro-c, the famous enro-c, and was going to be presented by my wife, which is not feeling well. Her throat is sore, and she asked me to do this quick presentation. This is the aspect of an hydrogel made up with alginate, sodium alginate. We are trying to patent the way we polymerize the alginate. We add enro-c and some propylene glycol. We end up with a gel that, to our surprise, is fantastic in terms of healing many types of wounds. And we have, just for cattle, but we have used these in many cases in all animals. And I'm also a physician, and I can tell you that I have used it in some human patients. It prevents new calves navel infections based on the biocompatibility of alginate and enro-c. We use it as a remedy to treat pink eye, treatment of teeth cracks and fistulas and lesions in dairy cattle, and even in a very challenging clinical case of burned animals. So you'll see it in a moment. Okay. To see if it really worked, one of the problems that are not well identified by many farmers is the stump of the navel that remains after calving. And what happens is that we challenge enro-c against iodine preparation, commercial preparation of iodine, to see which one of them work better. And you can see that it's very easily smeared into the stump, and according to humans, it feels a soothing effect, refreshing, which is very useful for a quick regression of the stump and resolution within 24, 48 hours at the very most. So detachment of this occurs within 30 days in all calves that we treated, something like 200 and something, and that didn't happen when the iodine preparation was used. In the first case, we needed only one smearing, at the very most twice, but the animal remained almost clean and with no problems, no infection. And with iodine, more than 3 applications were needed, and 25 umbilical infections or abscesses were registered. This has been already published, but the farmers are already asking for the product. It's not yet available, but my wife has a quick preparation scheme at the University of Mexico for those who need it. And also we tested pink eye with a higher concentration of enro-c, And we tested it against the standard, one of the standard treatments of pink eye in Mexico, which are both the topic and the parental administration of florfenicol. And according to this table, which I am not going to go into detail, we classify the severity of the lesions, but with enro-c alginate, the resolution was incredibly fast because of the healing ability of alginate, which is also used in human beings. Not this preparation, but other alginate preparations. And with enro-c, we got rid of moraxella very quickly, and treatment was noticeably cheaper in terms or by comparison with these two other remedies available in Mexico. Okay, this is the challenging problem we had at the University of Mexico. A barn got fire and the roof fell into these cows, and there were 3 of them, and as you can see, the burn was very severe and almost in the whole body, and they were treated medically to support with analgesics and psilocin and so forth. But what really cured them, and as you can see, it was a challenging problem, was alginate in the preparation that we manufacture at the laboratory in Mexico. Little by little, the lesions were cured, and around 3 weeks or 4 weeks, we ended up with an animal that it was almost cured and finally got cured. One cow was sacrificed, was put down because he was so severe, and we were aware that he was suffering. So from the 3 animals, 2 recovered fully, and to recover those type of burns in the field is not easily at all. This is just a quick presentation of the incredible capacity, healing capacity of this new gel that we are about to put into the market in Mexico if the company manufactures it. I don't know if there is any any questions about the gel or anything that you want to mention.
I'm just curious, did you ever try just using the hydrogel without enrofloxacin? Not for the burn cases, but for milder things like maybe the navel, or others?
Yeah, and alginate, sodium alginate and other alginates are being used in human medicine.
Yes, yeah, that's
and they work fantastic.
Yeah.
But the combination in veterinary medicine prevents many infections because, you know, animals are in the barn or in the field, and they're usually full of flies.
Right.
So
Right.
that prevents further infection. But that's
I was just
very
curious
interesting.
if you use that, because enrofloxacin is one antibiotic which is used everywhere. It is possible that we may have to incorporate a different antibiotic in this alginate for certain conditions, not everything. That's why I thought I will ask you whether hydrogel itself, without any antibiotic.
We have tested it,
Yeah.
and it works beautifully.
Yes.
Okay. Well, if there are no further questions or observations, I would like to call Dr. Swinkels. He will talk about the effect of additional prednisolone in intramammary treatment of clinical mastitis. This is a very interesting subject because little has been written about this subject. So let's hear.
Thank you for this introduction.
To advance and to point out.
Yeah. Thank you for this introduction. I thank the organization for the opportunity to present this, I agree, exciting study. It's a study that was done in France with MSD-ML Health, the clinical research organization CEBIPHAR, and the University of Clermont-Auvergne, and Vet Agro Sup. This is about clinical mastitis, and it is about the additional treatment with prednisolone. So if we see in Europe, the most frequently sold intramammary tubes have antibiotic and prednisolone. The top two products are like that. Specifically in France, almost 60 to 75% of the cows with clinical mastitis are treated with antibiotics and prednisolone. We did an experimental trial in Cornell in 2013. You can read that in Journal of Dairy Science. And there we could see that prednisolone completely blocked the inflammation, but that is an experimental trial. So then you treat with additional prednisolone 4 hours after the experimental infection, which is not at all in line with what happens in the field, where farmers discover the clinical mastitis relatively late and where treatment is much later in the inflammatory cascade. So interestingly, despite the frequent use of additional prednisolone, nobody ever did a field trial if this has any value. Everybody assumes it works, but nobody ever showed it. So that's why we thought we need to do this trial. It's evidence-based medicine. That's what we as veterinarians would try to aim for, and that's why we want to show the effect of prednisolone. The problem with additional prednisolone, showing that in the field, is that clinical evaluation is subjective. And also, there's a lot of variation between animals in clinical effects. And that makes a field trial very difficult. So there is a reason there are no field trials. But technology is developing, and Lely robots, they have milk sensors and they have cow sensors. So we can objectively measure what happens in the milk, and we can objectively measure what happens in cow behavior. And we can also measure it very frequently, and that is an advantage. And we thought maybe if we do that, if we use these sensors, then we can show the effect. So that was our objective, to show the effect of prednisolone using cow and milk sensors in Lely robots. So we selected 9 French farms in the area of Bretagne, Brittany. They all had to have an A4 or A5 Lely robot. They had to have the cow sensors installed, the SCR sensors, and milk sensors. And the milk sensors, they measure conductivity at the quarter level. They measure somatic cell count at the cow level. This is a California mastitis test that is inline, so that's automated. And they measure milk production at the cow level at every milking. And the cow sensors measures rumination and activity very frequently at each cow, and during, they can measure it every 2 minutes. Although the Lely robot could only give us the data, the 24-hour average, which was a pity, but that was also, we used that. So these are the 9 farms. We added 3 extra farms to get more cases because they are low somatic cell count farms and they didn't have a lot of clinical mastitis, so we added farms to get more cases. They all had the Lely A4 or A5 robot. And, well, the average number of cows on a farm in France is 70, 75 cows, so these were above average dairy farms. So in the Lely robot, we have a Horizon or T4C software, and they produce 2 reports related to mastitis, report 12 and report 23. And report 12 is an algorithm, is based on an algorithm measuring drop in milk production, conductivity increase, somatic cell count, and it combines all those data, and then on the screen it says, this cow has mastitis. Please check the cow. And this is very reliable, 95% that the software says this cow has clinical mastitis. It is indeed a clinical mastitis. So farmers value this very much. So that's what we used. And that report was checked twice a day by the farmers. Then they had to go to the cow to confirm the clinical case. And then they would call the local veterinarian. I'll explain why we needed to do that. And we only included mild and moderate mastitis. That was because farmers wanted to also use additional parenteral treatment when symptoms became severe. But this is 80 to 85% of the clinical cases anyway. They are mild and moderate. Yeah, so the treatment was twice a day for 10 to 12 hours interval for 2 days, and we had 2 tubes, one without prednisolone, with an antibiotic, and one with the antibiotic and prednisolone. This is a commercial product, but this tube had to be specifically produced to be exactly the same as this one, but then only the prednisolone was taken out. The rest was all in, all the carriers, everything else was in there. You see that the tubes are exactly the same. So the farmer and the vet, they didn't know what they were treating with. So here you see a little A, you cannot read it, but this is a little A and there's a B. So they could read A and B, but they didn't know which one was what. So they were completely blinded and the treatment was randomized. So just before treatment, the veterinarian would take samples for bacteriology. He would take a milk sample for cortisol. He would do a clinical examination, and the somatic cell count data came from the automatic milking system. The next 3 treatments, the farmer took over. He took samples for cortisol in milk. He did a clinical examination and at every treatment. Then the farmer, independently of this, observed specific behavior related to pain. I'll come back to that later. He did that for 2 times a day for 4 days. Then, of course, the robot measured conductivity, somatic cell count, as we discussed, at every milking. And the sensor here measured rumination and behavior. And after 14 days, the farmer did bacteriology. So, as we expected, we compared the 2 treatments. We have 35 cases in this group, 36. We aimed for 40. We calculated we needed 40. So the trial has not completely finished. These are preliminary data. And as you expect, there's no difference between the groups in the bacteriological cure, which was 77 in the non-prednisolone group and 82 in the prednisolone group. So there was no difference, and that's what you expect. Typical for mild and moderate mastitis, we found CNS, we found negative cultures a lot, and we found this is what happens sometimes. Even if you randomize, you find 28 cases of Enterobacteriaceae in one group and only 3 in the other one. This is very unlikely that this happens. This was mostly E. coli. unlikely to happen, but it can occur. And strep uterus, we found that as well. So you see mainly environmental mastitis and a lot of CNS. So here we see an interesting result. The concentration of cortisol in the milk at every treatment. So treatment 1, 2, 3, 4. The dots are the cows. So we had 35 cases or more or less in each group. And the green ones are from the cows that were treated without prednisolone, with antibiotic, but without prednisolone. And the red ones are from the cows treated with prednisolone. And of course, you see the variation in cortisol levels in cows. And we found a significant difference at the second treatment between no prednisolone and prednisolone, which is the first time that that has been shown in the field. And this means improvement of animal welfare. Yeah, so cortisol is a stress hormone. And if you can reduce that, is a sign of improved animal welfare. At the other time points, we didn't find any difference, only at treatment point number 2, so after the first treatment. This is the effect on somatic cell count. Yeah, so this is the somatic cell count from the robot, from the automatic milking system based on a CMT, an automated CMT test. This is the Kaplan-Meier survival curve. So the line going down means the somatic cell count is dropping below 200,000. Yeah, so it takes 7 days to go below 200,000 for cows treated with prednisolone, and it takes 12 days to go down below 200,000 for cows without prednisolone. That was significant. The other effect was on activity. This is the situation from day -14, so that's 14 days before the clinical mastitis occurred. That's the advantage of the sensors. They get continuously monitored, so you have also historical data. And you could just monitor 14 days before, and we saw already activity going down 4 days before the farmer discovered the mastitis. Yeah, so imagine that the system discovers the mastitis already 4 days before the farmer does. So we took -4 days as the cutoff, because then already activity went down. And here, after treatment, we took day 0, the day of treatment, as the starting point. And then we see that there was no difference before treatment and before the symptoms changed. But after treatment, we see a significant effect, meaning that if you treat with prednisolone, activity goes up. Yeah, so you can see it in the cows, they become more active. Yeah, this is interesting because there was this paper from de Boyer de Roche in 2017 and Ginger last year that were describing the specific behaviors of pain. If you really look at cows that are painful, you can see that on their standing posture, you can see that on their state of awakening, head posture, tail positioning, movement of tail and tail posture, and if they're eating. And we looked at differences there, but we found no difference. Okay, this is subjective. The farmer was scoring this. Yeah, so we found no difference, but this is the first time in the field that somebody looked at specific pain behaviors. So I think this is still quite nice, although we find no difference, but still quite nice. We can learn from this towards the future. We found no difference in milk conductivity at the quarter level, no difference in milk production, and no difference in rumination frequency. So milk conductivity is not a very reliable parameter. It goes everywhere. But there was a huge amount of variation. Here you see, just to give you an impression why we didn't find anything, This is the chaos. This is what happens before the clinical mastitis. Then it drops, and then it goes up. But you see, these are all the cows are in there, and you see the enormous variation. And that's why it's so difficult to find an effect in the field of additional prednisolone. We found no difference in rumination. That's what I would have expected. But the computer or the software was not able, it was only able to produce a 24-hour rolling average. So that means that the data were diluted. It measured rumination every 2 hours, but we only could retrieve 24-hour rolling average. That's why we found no difference, I think. Just discussion. Weak spots are that you just saw univariate analysis, but the prednisolone group got more E. coli than the non-prednisolone group. We still need to correct for that in the multivariable model. I don't know which way this will go. I don't know what the effect is, but we still need to do that. We saw a large variation, and we had low cow numbers, and that is because those farms, it's always the same when you do clinical trials. You have the best farmers, they don't have a lot of mastitis. And in France, you have small farms, and they don't have a lot of cows, and then you also don't have a lot of cases. So that was not easy, but still, the conclusion is that this is the first study that I am aware of that ever showed the effect of prednisolone in treatment of mastitis, while the majority of cows are treated with this. So I think this is nice to have these data. It improved milk quality, somatic cell count, cortisol levels, so animal welfare, but we didn't find a difference on rumination, milk production and conductivity, and on specific pain-related behavior. And the caution is that, yeah, preliminary data, but still, I think interesting enough and reliable enough to present. With that, I thank you for your attention.
Are there any questions? No? I have one. Have you done some studies of withdrawal time for prednisolone?
Prednisolone disappears very quickly. I think after 6 to 8 hours, concentration is already very low. So 6 hours after treatment.
Okay, so it absorbs or it is eliminated?
It goes into the cow. And it absorbs, yeah. It very quickly goes into the cow and gets metabolized.
Okay. I had the experience with the regulatory agency in Mexico of finding out a person that didn't want to accept the use of prednisolone in intramammary tubes. And that's why I made a huge search in data. And as you wisely said, there are no data about prednisolone except for one paper that I found in which they recommend the use of prednisolone, but just one from American Veterinary Research, I think. So this is
But was that a field trial?
It was a field trial, yeah.
Okay.
But as you said, it was all controlled and unnatural. And this is
It was an experimental trial, or?
Yeah, exactly.
But that's different.
This is a much interesting one because it adjusts to real life.
Yes.
Thank you very much. Any other question? No? Would you recommend to add prednisolone to other intramammary tubings?
Well, I mean, I would say if you see cortisol levels going down, and if you see the consumers becoming more critical of what we do in dairy farming, I would say yes.
Okay.
Yeah.
Okay.
And also because there are hardly any residues, no withdrawal time, and lengthening of withdrawal time. I'm in favor of it, yes.
Okay, it will also have an impact in the amount of healthy tissue for milking.
You would expect, well, so the effect of anti-inflammatory treatment, you would assume milk production would be better because you prevent tissue damage, but then you need to treat early in the inflammatory cascade. And in the field, we are relatively late, and the damage has already been done.
Yes.
That's why even showing this in the field is so difficult, and that's why I am, well, proud to see that still, if you treat relatively late, you see an effect.
Okay, one last question, because nobody is asking anything, but perhaps you were here when I was talking about enro-c, a new enrofloxacin entity.
Yeah.
And for chronic mastitis, would you recommend prednisolone? It will not reduce, for example, immune system or anything like that?
No, I wouldn't. I would not recommend it. I think it would have a very limited effect. Of course, the more severe the inflammation the more effect an anti-inflammatory. And the more earlier you are, the more effect an anti-inflammatory has. But if you go to chronic infections,
it's too late.
It's too late, and there's hardly a lot of severe clinical symptoms. So I think you will not find an effect.
Okay.
It will be very small. Yeah.
Good. Thank you very much.
Yeah. There's one.
I have one question. Have you tried the use of dexamethasone in an off-label usage, for example, because we have obviously both tubes with and without corticosteroids, prednisolone, but sometimes where we want to use, for example, a tube, it doesn't exist in the store, the shop, you know. So have you tried to use the dexamethasone that is used for intramuscular shot to put in there?
No. So first of all, there are no intramammary tubes with dexamethasone. And the injection, the issue with that is the dose is maybe 20 to 50-fold. Yeah? So this dose was only 20 milligrams. But if you look at dexamethasone, it's like 1 gram, or I mean, it's a huge dose. And a lot of cows with mastitis are pregnant. And so it's not practical to inject with dexamethasone.
No, I mean, obviously adjusting the dose, obviously, because it should be a lower dose because it's only 1/4 of the whole body.
No, but there are no commercially available tubes with dexamethasone.
I may comment on that, is that it's very dangerous, as Doctor pointed out. You may end up with a premature calving just by injecting 20 milligrams of dexamethasone will be enough to induce premature calving.
Yeah, excellent presentation, Dr. Sinkel. In the study, you use monitoring technology and you mentioned we will be able to detect before the onset of clinical signs. What do you think there'll be the ideal timing for treatment using technology? That the first treatment should be before, or just get to thoughts.
That is an interesting question. So you saw the farms that we used. Well, the modern dairy farm has a bulk tank in Europe, has a bulk tank somatic cell count below 200,000 at the moment, which means it's mostly environmental mastitis. And in environmental mastitis, there is a lot of self-cure. Yeah, so if you are 4 days before the farmer discovers, so you're in the early stages, if you treat, I think the treatment, especially with anti-inflammatory, will be huge. You will prevent tissue damage. Yeah, so I would be in favor to use anti-inflammatories in the early stage, but I would not be in favor to use antibiotics because of the high amount of self-cure. So I think if you go early stages, you don't know whether the animal's going to cure by itself or not. And I think you first need to study that you don't overtreat. Maybe you don't, but I think you need to study that, to what extent afterwards it was needed or not, to what extent there was self-cure or not. And I think we don't know that. But anti-inflammatory, I would be in favor. Yeah?
In Mexico, there are some compounds that contain only phytoelements, which do not need withdrawal time. For those cows that you're not sure whether they are going to go into clinical cases. So that may be another point to experiment.
Yeah.
Thank you very much, Dr. Winkles. Brilliant talk. Okay, now we are going to hear a presentation, evaluation of the treatment efficacy of generic enrofloxacin compared with pioneer enrofloxacin for the first treatment of naturally occurring bovine respiratory disease in commercial feedlot by Dr. Miles, I don't know how to pronounce, Theuer. Okay. You know this is to go forward, to point out something. Okay.
Thank you. Well, good morning, and thank you for coming for the presentation this morning. Happy to be here and appreciate the sponsors and the hospitality throughout all this entire event. So just to kind of give a little bit of conflict of interest disclosure, our company ourselves have provided consulting services for all of these up here, probably a few more that left off of. That's part of our role as a consulting veterinary group, act as a contract research organization. Always happy to add more companies on there as we go forward as well. And for the trial that I'm presenting here today, Virbac Corporation provided funding for this. However, none of the employees were involved in the handling, executing, or analysis of the study. So just to give you a little bit brief background, so I'm part of veterinary research and consulting services from the United States. Primarily, if we're a feedlot consulting group, we do a little bit of dairy, calf ranch work as well. There's 12 of us veterinarians that work together as a think tank cooperative standpoint, 70 feed yards from Texas to Washington. So every day, 2 million head of cattle on feed. And so our whole practice is really based upon evidence-based medicine. So before we put tools and protocols out into the field, we want to generate some data to make sure we know how it's going to work for our clients. So we had a great presentation this morning about some enrofloxacin standpoint. And so from the United States, it was first approved in 1988 as the pioneer product called Baytril 100. Bayer Animal Health was the one that brought that to market for us in the United States. And then in 2002, a generic version of the pioneer product named Tenatril injectable solution was granted a bio-waiver. And so from the FDA, US FDA, active ingredient was the same formulation in Pioneer. However, one of the great presentation this morning side was looking about, so some of those generics, the pharmacodynamics may not be quite all the same. And so we wanted to have some field data comparing the generic to the Pioneer product for adoption to the field. So our study and objective was to compare the treatment efficacy of Tenatril injectable solution to Baytril 100 for first treatment of bovine respiratory disease in a commercial feed yard in Kansas. And our secondary objective was to evaluate the health outcomes of traditional beef cross cattle compared to dairy beef cross. Here in the United States, the use of the sex semen onto the dairies, breeding those to the dairies, cows to beef bulls has resulted in a great magnitude of our cattle supply. And so an opportunity to, as we learn how to manage these cattle in the feed yard, to evaluate some subsequent health outcomes. So the feed yard, which we conducted this study, averaged just a little over 31,000 head during the time period. We began enrolling in end of August of 22, and we followed these for 60 days, and have completed the first part of January of 2023. So for inclusion criteria, this would be pretty typical in our situation standpoint. Be pen rider personnel trained by feed yard managers or consulting veterinarians to identify sickness. And there on the right will be some of the signs that we see. We know it's a very inaccurate science standpoint, but that is kind of the best tools and technologies that we have today. And we talk about the previous presentation about some of these technologies. Unfortunately, we haven't been able to get those adapted into the feed yard aspect yet from a cost and actual data and accuracy. A lot of things are coming in the pipeline. So looking for our clinical illness score, anywhere from 0 to 4, with 0 would be our bright alert, healthy, to 4 would be the open mouth, morbid. Those would be animals that would be euthanized. And so our inclusion criteria would be anywhere from 1 to 3 from that severity standpoint. And so these cattle had to be no previous treatments. Rectal temperature had to be greater than or equal to 40 degrees Celsius. Had to have at least 60 days to harvest. And then they were visually observed by myself or trained personnel for no other signs in any other organ systems. So enrolled these into a 1-to-1 ratio within lot. And so that way from the feed yard side, so as we manage things from the lot level randomization, those origins, days on ration transition, where they are in the feeding cycle, all those things, we controlled for that within that lot level randomization. And so, 4.5 mgs per pound there, so 9.92. Each dose was the same for the tinatril versus Baytril. Then we caught body weight and rectal temperature, and we captured those in our computer health systems. So we took a total of 500 head and enrolled them at lot level. So we got 250 each way, average 6.67 enrollments per day with a median of 5. It ranged anywhere from 0 to 30 enrollments a day. So pretty typical enrollments and pulls from a commercial feed yard, especially in the fall, with the amount of variation standpoint. But that's how long it took us from an enrollment side. So prior to a study completing, a total of 7 calves were removed from the study prior to analysis. So these were due to things subsequently pulled for foot rot, died due to digestive issues, lameness, a bular injury. So these were things that myself as the investigator were not attributed to the treatment group. So we had 5 of the tinatril and then 2 from the Baytril. So you'll see from the subsequent analysis will be with 493. This is kind of further describing some of that population side from the commercial feed yard, especially from in the United States. 437 of those were not administered metaphylaxis. Still a practice of what we do, especially on moderate and high risk populations to manage disease. So we're still trying to use that very judiciously. 56 of those calves were administered telithromycin, so tolocin 100 upon arrival processing. Kind of further breakdown that other group, so 352 of these calves were traditional beef breeds, and then 141 of them were dairy beef crosses. So upon enrollment, they were returned back to their home pen, and we monitored them back there for 60 days by pen rider personnel, blinded a treatment group. The treatment administration was different from the pen rider group. And so they were, pen riders were able to pull those cattle and bring them back. We used, if they identified the clinical signs for BRD, they were retreated. If they exceeded the PTI, the post-treatment interval period, we used a minimum of 3 days before any calves were eligible for retreatment. And so if they were identified by the pen writer and they either had a rectal temperature still greater than 40 degrees Celsius or lost body weight, they were automatically retreated. And then if they displayed severe clinical scores, they were retreated regardless over body weight or rectal temperatures if they were pulled back. And so we also follow these all out for a minimum of 60 days. So no animals were removed from study for being marketed early or moved to regular practices. From a stats analysis standpoint, so everything from binary outcomes were created for treatment success and case fatality risks, and we use linear mixed models to account for functions with the outcome variable, so Gaussian distribution for our continuous outcomes, binomial models for dichotomous, and then our cumulative logistics for our ordinal outcomes, which would be our clinical illness scores. We included lot level as a random intercept for all animal level models to account for the clustering. So this kind of set up this table here as far as the kind of set up here with whales. So these tables will be here on the left side will be some of the outcomes. So I can make quite formatting wise, and then so tenatril would be here on the left, and then batril would be here on the right. And so basically these cattle, we were about average 58 days on feed, 390 kigs out of enrollment. So pretty well typical for our United States, typical more from a yearling processing cattle standpoint. Rectal temperatures, no differences upon any of those enrollments. So just kind of further describe, so you see a breakdown over how those clinical illness scores there, no difference there between treatment groups, which was great from the design standpoint. We were, pretty well from 1s to 2s would be predominantly for those clinical illness scores descriptions. And then you kind of break down, 88% of those cattle, no metaphylaxis, and then we can see here 29% of those cattle were the dairy beef crosses, with the remainder being traditional beef breeds. So some of the highlights, our main take-homes, really looked about it here. So on the x-axis would be the first treatment success there on the left, on your left, and the case fatality risk be on the right. And so when we looked at the first treatment success, that was defined as not being treated additionally for bovine respiratory disease and not dying due to bovine respiratory disease. And then case fatality risk would be those cattle which died due to bovine respiratory disease as part of the trial. And so saw no difference, so 0.19 between treatment groups, numerically just a slight increase from tenatril, but again, no statistical difference, and then no difference from case fatality risk. And so, those were the primary outcomes what we were looking at for this study. So really kind of showed we saw no differences between those two treatment groups. Break out some more of the detail here, comparing out the two trial treatment groups. Seeing there from the second treatment success, we're at 55%. So we would have been, the treatment regimen would have been, florfenicol, and then an oxytetracycline would have been the third treatment group for these cattle. And so looking at the third treatment success, or case fatality there, we saw the treatment death interval was, we're about 12 days, 12.8 days for tenatril versus 18.4 for the batril, but the p-value is 0.11. So enrollment description by cattle type. So looking at some of these, breaking out, and looks like the format got messed up, so I apologize. Some of these, the days on feed, at enrollment, we'll see really not different wise. However, the enrollment body weight, sitting here, was 339 versus, in the dairy beef crosses versus 403 kgs for the traditional beef breed. And so our dairy beef crosses were lighter at time of enrollment, which would be pretty typical from our production systems. We'll see those cattle come into the feed yards a little lighter in weights. Some of those are coming straight from the calf ranch at 3 or 350 pounds, and some of them are going through a backgrounding phase and coming at 650 or 7 weights. So some variety, but as a whole, we'll typically see those lighter than what we see the traditional beef breeds cattle. No difference there from rectal temperatures or clinical illness scores as well between treatment groups. As we go through from health outcomes, one of the things to really kind of take apart from this, we saw no difference in these health outcomes for first treatment success or case fatality. And so this differed from some of the research that we've done before. Looking at these is kind of looking at these subpopulations and opportunities where we typically see a more challenge, different type of challenge in the dairy beef crosses. Sometimes we'll see those will continue to die throughout the feeding period. You know, and a lot of them more from other diagnosis. Some of the times it seems to be more tied to source specific for us in our hands and observations. So in previous research we've done, we've seen poor health response with these, and however, that was also confounded by lighter body weight. And so I say as a whole, we bring this more to attention because as we look at our U.S. cattle supply, these are representing about 20 to 25% of the cattle on feed that we work with on a daily basis. And so more research, we need to figure out how to manage these dairy beef crosses most effective to maximize their potential. And I think it makes a great model and a great calf, but there's still things that we need to figure out how to manage these most effectively. So in conclusions, there was no statistical difference in health outcomes in cattle which were administered Tenatril injectable solution compared to Baytril 100 for first treatment of bovine respiratory disease in a commercial feed yard. And so having these data in hand, practitioners should be able to use these products interchangeably based upon the results of the study. And so it's been great to be able to have that data to be able to show that to our feed yard managers, our personnel, to say, here's some of the reasons why, so now we can figure out the most cost-effective solution for our clients. So all of these data are published in the Bovine Practitioner. Put a plug in here for the Bovine Practitioner is from academics and researchers at all standpoints. A great place to submit publications. We are an open access journal and no fees for publication. So subjectively, I am one of the current associate editors there, so we're always recruiting projects. So if anyone has any questions with that, don't hesitate to reach out. And it's kind of from a safety situation for Tenatril and enrofloxacin. You can use the QR code there to get the full prescribing information. For our hands, it's 28-day withdrawals, and we can't use it in dairy cattle older than 20 months of age. But with that, I'd be happy to answer any questions anyone may have.
It's just a question about how you have the scoring system there. I might have missed it, but I did not see any respiratory score there. Like you have, like 1, 2, 3, 4. Yeah, that one. Usually the depression, depression score. And what about respiratory? Isn't that an important?
So when you look at a lot of these clinical illness scoring standpoints, you can add a lot more things in there. However, at the end of the day, they are subjective assessments standpoint, and you got to draw categories with those. And so we're trying to kind of do things at the speed of commerce. And so some of those things with the depression, will be in there. The respiratory sign will be one of the ones that the feed yard people are pulling as part of the pen rider standpoint. So that's what they're looking for. And this clinical illness score was more assigned at the shoot level standpoint.
Okay. Okay. Thank you.
Thank you for your presentation. There was a very significant difference in the body weights of the groups. So group T was about 60 kilograms lighter, and so they received much more enrofloxacin than the other group. Do you think that these affect your results?
So the question was about the difference in body weight, so receive different amount of enrofloxacin when it affect results. And I would say a great comment, always have confounded with that, especially when we're dosing the body weight, and that's one of the great things that we get to do in veterinary medicine versus our human colleagues wise. So I think when we look at those, interesting to see from the PK/PD responses wise. I think I don't think the short answer is I don't believe it would affect the results, because we look at that, we're dosing that animal to that range that it needs, and also we have a range in the United States for that, was brought up earlier, for the doses over for enrofloxacin, and we were at the 4.5 mg, so we're kind of in that mid standpoint.
But the difference in the body weights didn't only present that the dosage is different, but also that the ages of the animals are different.
Correct. Yep. So age of animals would be different as well. That would be correct. And so, in our traditional beef breed, unfortunately, we do not, in the United States, have a full traceable system standpoint. And so, when we are in the feed yard, we're looking at body weight. That's what we know. That's what we can rely on. Something that maybe over time we can continue to move forward. But age and body weight are confounded as far as the way that those animals are raised throughout the entire system. So.
Any other question?
Well, thanks for the presentation. Just a question, in your experience, did you measure at any time body weight gain and lung scores at slaughter with these products?
So the question was related, did we measure body weight gain or lung lesions at slaughter for this treatment trial? And no, we did not. We've done body weights historically on these field trials, looking at them, and we find out that it's not all over the place. Now you're having to go through into home pens to sort cattle back out, messing with the social structure, and by the time you get them to pens to catch body weight, how accurate is that? I think it provides, it's an objective measure, but however, I'm not sure the juice is worth the squeeze when it comes down to it. When we look at these, we're looking at those cattle that had to be subsequently retreated or died, as that's where the rubber meets the road. The idea more for the prevention, if we're going to really improve the bottom line of the feed yard, identifying efforts over how to prevent them from getting too sick versus which one responded better has been more effective in our hands.
Well, I have an observation. Enrofloxacin has become a very cheap raw material, and producers, maybe China, India, and so forth, they could produce an incredible high-quality raw material, but they will also produce a low-quality raw material. So in the end, the question here, seeing that results were not statistically different, in the end, the farmer will ask which one is the cheapest. Could you comment on that?
So yeah, the idea that the price and all of the generics are created equal, I think is very questionable side. I think the idea, having one of the things that's generating that data, whether it's your own data that you do internally to validate before you use those products, or some evaluating the fields to make sure that those data, those generics are all the same. And there's great rules and regulations that they have to jump through. So a lot of companies, it's not like they're just handed that from at least from the U.S. from the FDA regulations. But I think the idea of having that field data to validate that model is most effective to make sure that you're getting the viable product and outcomes what you expect.
Okay, thank you very much. Any other question? No? Okay, we thank Dr. Miles for his good presentation. Beautiful, thank you. Finally, we have an additional presentation by Dr. Paulo Henrique Jorge da Cunha, do Brazil. The effect of metaphylactic use of tildipirocin for the control of naturally occurring bovine respiratory disease on performance and probability in high-risk feedlot cattle. Thank you very much. Este es para to advance and to point out something. The light.
Okay, thank you. Good morning. I would like to thank the committee for the opportunity to speak today. My name is Paulo Cunha. I'm professor at Universidade Federal de Goiás from Brazil. First, I would like to present some data about Brazilian livestock. Brazil has the oldest, largest commercial cattle herd, and it's estimated in 200 million herd. Brazil is divided in 5 regions. Center-West has the largest number of cattle herds with 34%. The second region is North with 24%. But this second region will be difficult to increase the quantity of animals because it's Amazon region. So because of the laws, it's difficult to create new areas to form pasturing. Then the Northwest with 13, Southwest with 17%, and South with 11%. In this graph, we can see the percentage of feedlot per state. São Paulo with 34%, Goiás almost 26%, Mato Grosso 20%, Minas Gerais 11%, and Mato Grosso do Sul 8%. Professor Danilo Millian from Unesp, he invited 54 constant feedlot nutritions to participate in the survey. These nutritions were selected because they represent typical cattle feeding present in different geographic locations of Brazil, and just 36 complete the survey. In this table, we will focus just on the category of bulls, because it's the major category that we can find in feedlots in Brazil. So the average initial age is 36 months. The average initial body weight is 376 kilos. The average final body weight is 555. Days on feet, 106. And AG, almost 1.6. In this graph, we can see the distribution between feedlot cattle at slaughter and no feedlot cattle at slaughter. So feedlot cattle, it's almost 70%, and no feedlot cattle, 82%. We believe that in the next years, we will increase the number of feedlots, and consequently, we will increase the health problems. We made a research in a feedlot in the state of Goiás with 3,120 confined cattle. BRD morbidity was 1.12%, and most of cases of BRD were confirmed in the first 3 weeks of days on feed. So BRD is the most prevalent disorder in feedlot in cattle, and BRD is a complex multifactorial disease involving interaction between environment, host, agents, and bacteria. One of the problems is cattle with disease may not always demonstrate declining signs that are characteristic of BRD. One example of this was involved in our study, the slaughter of 1,900 cattle was monitored, and 380 lungs, 6.82%, were selected with microscopic alterations. Among these samples, 55 confirmed pneumonia with histopathological exams. So the big challenge of BRD is an early diagnosis and treatment. One of the tools to reduce BRD risk is metaphylaxis. Metaphylaxis is the practice of administering an approved antimicrobial to an entire lot or pen of cattle with the intent of controlling the incidence of BRD in the cattle at significant risk for BRD. The aim of this study was to evaluate the impact of metaphylaxis on morbidity, average daily gain, carcass weight, processing cost, revenue, and profits of high-risk cattle of BRD in feedlot. We acquired 208 high-risk crossbred bulls for a commercial feedlot farm. The animals were divided in two groups. One group received metaphylaxis up arriving, and the second group did not receive metaphylaxis. The first group, we call it MET group, with 104 animals. They received a single subcutaneous administration of 2-dipyrosine at a dose of 4 milligrams per kilo body weight. And the second group, called control group, with the same quantity, received a subcutaneous administration of 0.9% saline at the same dose. Both groups have almost the same average body weight, 306 kilograms. Both groups were processed and received vaccine for Clostridial disease, BRD vaccine, and oral antiemetics, and the days on feed were 94 days. After this period, all the animals went to the slaughterhouse. We analyzed the lungs. Lungs with lesions, we collect samples for histopathological and PCR tests. We received recently these results, so unfortunately, it will be not possible to share today these results. One important point is the criteria to inclusion in one feedlot, the metaphylaxis strategy. The inclusion criteria for this research was animals with high-risk cattle, external source acquisition, body condition score of 1 and 2, considering a scale from 1 to 5. The feedlot, we said that the animals had poor body condition. And the last criteria was the distance traveled more than 5 kilometers and doing more than 8 hours with 3 days of rest on arrival at the feedlot. The animals, they traveled almost 800 kilometers to arrive to the feedlot, and the time that they spent was almost 10 hours. The diagnosis of BRD was made in 2 steps. The first step, pen riders identified the animal sick with nasal secretion, weight loss, and apathy. We did not measure fever because the temperature of this time was very high. And then, in the second step, the veterinary of the feedlot established the diagnosis of BRD by physical and hematological examinations. The results of clinical, subclinical, and the hematologic results we published in an abstract of this Congress. The statistical analysis was the data was performed using the ANOVA test. Student t-test was applied to the average daily gain, carcass weight variables with a 5% significant level using the R software. In this graph, we can see the average daily gain. The average daily gain for the meta group was 1.82, and the control group was 1.67. The average daily gain was statistically significant based on the p-value. In this second graph, we can see the carcass weight. The carcass weight for the meta groups was 288, and the control group was 208, and the carcass weight was significantly different considering the p-value. Metaphylaxis increased the processing cost. Meta group was almost 45 reais and 52 cents, which is equivalent almost $9, and the control group was 9 reais and 42 cents, which is equivalent almost $2. The costs were compensated by increased revenues and profits of meta group versus the control group. The revenue is 3.14%, and the profits were 11%. The difference in performance and profitability between the two groups could be explained by a lower BRD morbidity rate in the meta group compared to the control group. In this graph, we can see the bovine respiratory morbidity rate in meta group with 8.6% and the control group 23%. In conclusion, this study demonstrates the advantage of antimicrobial metaphylaxis with tioprosin in high-risk cattle on their arrival at the feedlot in reducing BRD morbidity and improve average daily gain, carcass weight, and profitability. I would like to thank Dr. Salmi and Dr. Alfieri from Universidade Estadual de Londrina. They were responsible for the histopathological and PCR exams. And the feedlot, Nutritaurus, they opened the door for us to do this research, and MSD for the foundation. Thank you for your attention. If there are any questions, it will be a pleasure to answer.
Any comments or questions? Please. I have a comment. The World Health Organization has encouraged all the countries worldwide to take care of the way we use antibiotics in food production. In my view, I agree completely with your results, but in my view, we have a problem in Mexico. Our authorities tend to forbid the use of the word metaphylactic or prevention in the information when you want to register a new product. Have you any comment on the impact of using tioprosin in human bacterial-resistant data?
I think that we would totally agree. We need to take more care about use this kind of strategy. We know that there is a relation between this kind of strategy and the possibility to increase the antibiotic resistance. But when owners of feedlot, in Brazil, they intend to use this kind of strategy, I think that there are two questions that we must ask them. First, the animals are in the high risk of BRD. I think this is one point. And the second point, the price. When we did this research, the owner received from the slaughterhouse $4 per kilo. Nowadays, if we repeat this research or an owner wants to use this, they will receive $2.06. So I think that now the big challenge is the economic situation. In Brazil, we are now in a bad economic situation, so the price of the kills that the owner will receive, it's in a bad moment. So I think that we have to take care about this. We just can use this strategy for animals with high risk, and it's important the owner of the feedlot to know that it's not to use in any situation. There is a specific situation that we can use. I made a research, not in cattle, but in small animals, and some owners of the small animals, they have problems with the resistant bacteria, with the same antibiotics that the dogs were receiving. So I think that we have this problem, not just with beef cattle, but in all kinds of animals, like pets too. So nowadays, I think that it's an important strategy, but we have to discuss a lot with the owner of the feedlot, with this important to do this at this moment.
Okay, thank you very much. Any other question or comment? I just have one final comment, is that we humans are a lot in this planet, and the real plague are us. We are 8,000,100,000 people in the world. Treating animals is hardly a problem as far as I can see in terms of bacterial resistance, but regulatory agencies are tightened the requisites to register a drug. We must join efforts to clarify the World Health Organization that producing food is essential for humankind. Thank you very much for your attention. Thank you, Doctor.
Thank you.